In 2022, a team of geneticists published a landmark study based on ancient DNA extracted from a medieval Jewish cemetery in the German city of Erfurt. The remains have been sealed underground since 1349, untouched, uncontaminated, perfectly preserved through nearly 7 centuries of European history. When the lab results came back, they didn't just answer one question. They answered the question that has divided historians, theologians, and geneticists for over 100 years. Where did the Ashkenazi Jews actually come from? That question matters for reasons that go far beyond academic curiosity. The Ashkenazi Jewish community, the Jewish population that historically settled in Central and Eastern Europe, has produced a remarkable concentration of scientists, philosophers, musicians, and writers relative to its size. Albert Einstein, Sigmund Freud, Franz Kafka, Bob Dylan. The list is long and disproportionate in ways that have generated as much controversy as admiration. But before we can understand who these people are, we have to understand where they came from. And for over a century, that origin story has been genuinely contested. There are 2 main competing frameworks. The first says that Ashkenazi Jews are the biological descendants of ancient Israelites who were expelled from their homeland during the Roman period. First in 70 AD, when the Second Temple was destroyed, and again after the Bar Kokhba revolt of 135 AD. Under this framework, they are a Middle Eastern people who carried their culture, their religion, and their biological heritage westward into Europe across 2,000 years of displacement. The second framework, known as the Khazar hypothesis, argues something completely different. It proposes that Ashkenazi Jews are not primarily descended from ancient Israelites at all, but rather from the Khazars, a Turkic people who established a powerful empire in the region between the Black Sea and the Caspian Sea during the 7th to 10th centuries AD, and who converted to Judaism as a state religion. Under this hypothesis, Ashkenazi Jews are genetically Turkic, not Semitic, and their Middle Eastern identity is cultural rather than biological. The Erfurt DNA was about to resolve this debate in a way that no amount of historical argument ever could. There is a reason this question matters beyond the academic. The Ashkenazi Jewish community, numbering roughly 11 million people globally, is among the most genetically studied populations on Earth. That study has generated data precise enough to detect population movements that happened 2,000 years ago. It has also generated findings that challenge comfortable assumptions on multiple sides of the historical debate. The Erfurt cemetery was the key that finally opened the archive. The city of Erfurt sits in the heart of what is now the German state of Thuringia. In the medieval period, it was one of Central Europe's most significant trading centers, a prosperous market city at the intersection of major commercial routes, with a substantial Jewish community that had lived there for generations, operating as merchants, moneylenders, and skilled craftspeople within the heavily regulated framework that medieval European law imposed on Jewish residents. On the night of March 21st, 1349, the Jewish community of Erfurt was destroyed. The killing was part of a wave of violence that swept through Jewish communities across Germany, France, and the Low Countries in the wake of the Black Death. As the plague devastated European populations, and no explanation could be found for it within the scientific or theological frameworks of the time, Jewish communities were blamed. In Erfurt and hundreds of other cities, the accusations triggered organized slaughter. The Jewish cemetery of Erfurt was sealed after the events of 1349. The site was not rediscovered until the 20th century, when construction work in the city uncovered the remains of dozens of individuals buried there. The site was carefully excavated, and the skeletal remains were preserved for scientific study. What made the Erfurt cemetery uniquely valuable for genetic research was precisely its sealed history. The bodies buried there had been placed before 1349, and never disturbed afterward. This meant the DNA inside their bones, specifically inside the dense petrous bone of the inner ear, which resists decomposition better than any other skeletal element, represented Jewish genetic heritage from before the major population disruptions of the 14th century onward. This was, in the language of genetics, a pre-bottleneck sample, a window into the Ashkenazi genome before the catastrophic population collapses of the late medieval and early modern period compressed it into the narrow genetic profile we see today. The extraction team from the Max Planck Institute for Evolutionary Anthropology worked with samples from over 30 individuals recovered from the site. The results, published in the journal Cell in 2022, were definitive. The first major finding from the Erfurt ancient DNA study confirmed what many geneticists had already suspected from studies of modern Ashkenazi populations, but never been able to verify with pre-medieval ancient DNA. The paternal lineage of Ashkenazi Jews traces clearly and consistently to the ancient Levant. The Y chromosome, the genetic material passed from father to son, of the Erfurt individuals showed haplogroups and genetic patterns characteristic of ancient populations from the region of modern Israel, Palestine, Lebanon, and Syria. This is the area that ancient sources called the Levant, and that the Jewish tradition identifies as the historical homeland of the Israelite people. The data confirmed that the male line ancestors of these 14th century German Jews had indeed originated in the Middle East. The religious and historical tradition that placed Jewish origins in the ancient Near East was not mythology. It was genetically accurate. But the story did not stop there. Because the mitochondrial DNA, the genetic material passed exclusively from mother to child and used to trace female line ancestry, told a more complicated story. The mitochondrial profiles of the Erfurt individuals did not look primarily Middle Eastern. They looked European. Specifically, they closely matched haplogroups common in southern European populations, particularly Italian and, to a lesser extent, Greek and broader Mediterranean European women. This finding matched what earlier studies of modern Ashkenazi populations had suggested, but not conclusively proven. The most likely historical explanation: when Jewish men migrated westward from the Levant into the Roman Empire during the 1st and 2nd centuries AD, they settled first in Rome and the major cities of Italy, where Jewish communities became well established. Over time, some of these men married local Italian women who converted to Judaism. Those women's mitochondrial DNA, their European female line genetics, entered the Ashkenazi population and has been carried forward by every generation of Ashkenazi women since. The Ashkenazi founding population was, from its very beginning, a hybrid. Middle Eastern men, Southern European women, and a shared religious and cultural framework that held the combined community together and defined it as Jewish across every subsequent generation. The Khazar hypothesis has had a long and politically charged life. The Khazars were a real people, a semi-nomadic Turkic confederation that controlled a territory roughly corresponding to modern southern Russia, Ukraine, and Kazakhstan. From approximately the 7th to the 10th century AD, the Khazar Khaganate was a genuine historical power, wealthy, militarily capable, and positioned at the intersection of the major trade routes connecting the Byzantine Empire, the Islamic Caliphate, and the Slavic peoples of Eastern Europe. The documented historical fact is that at some point in the 8th or 9th century, the Khazar ruling class adopted Judaism. This is attested in multiple independent historical sources, including correspondence between the Khazar king and a Spanish Jewish scholar named Hasdai ibn Shaprut, preserved in medieval manuscripts. The conversion appears to have been genuine and lasting, at least among the aristocratic class. The hypothesis built on this fact, that this conversion explains the origin of Ashkenazi Jewry, was articulated most influentially by the writer Arthur Koestler in his 1976 book, The 13th Tribe. Koestler argued that the Jewish communities of Eastern Europe were primarily descended from Khazar converts rather than from ancient Israelites. The implications were significant. If true, it would mean that most self-identified Jews had no biological connection to the ancient Middle East at all. The 2022 Erfurt ancient DNA study, combined with earlier genomic research, addressed this hypothesis directly. The findings were clear. Turkic ancestry, the genetic signal that would indicate descent from Khazar or related Central Asian steppe populations, is present in modern Ashkenazi DNA at very low levels. Estimates range from less than 3 to at most 5%. This is far below what would be expected if the Khazar population had been a significant founding group. It is the level of background admixture consistent with centuries of living in geographic proximity to Turkic populations without any major founding event. The Khazar hypothesis is not supported by the genetic evidence. The Erfurt data confirmed it. The ancient DNA from medieval German Jews showed a genetic profile consistent with Middle Eastern paternal ancestry and Southern European maternal ancestry, with no significant Turkic signal. The hypothesis persisted for so long not because the evidence supported it, but because it was politically useful to various parties for different reasons. The DNA ended that argument with the authority that only direct physical evidence can provide. Among all the findings from the Erfurt ancient DNA study, one of the most scientifically significant was what the data revealed about a period in Ashkenazi history that is sometimes called the genetic bottleneck. A genetic bottleneck occurs when a population is drastically reduced in size by famine, disease, conflict, or forced displacement, and then expands again from the survivors. The genetic consequences are profound and lasting. When a large, diverse population is suddenly compressed into a tiny group, most of the genetic variation that existed in the original population is lost. The small surviving group carries only the genetic variants that happen to be present among its members. When that small group subsequently expands into a larger population again, all the new individuals are descended from the survivors, and they carry only the variants the survivors had. The result is a population that is, by genetic standards, unusually similar to itself, as if everyone in it is at least distantly related, because in a meaningful biological sense, they are. Modern Ashkenazi Jews show exactly this pattern. Genetic studies have consistently found that the entire contemporary global Ashkenazi population, estimated at roughly 10 to 11 million people, appears to descend from a founding population of somewhere between 350 and 500 individuals who lived approximately 600 to 800 years ago. Every modern Ashkenazi person is, at the genetic level, approximately as similar to every other Ashkenazi person as fourth or fifth cousins from a typical non-bottlenecked population would be. The Erfurt individuals, buried in 1349, before the most severe period of bottlenecking, showed measurably higher genetic diversity than modern Ashkenazi populations. The comparison between the pre-bottleneck Erfurt sample and modern Ashkenazi genomes allowed researchers to date and quantify the bottleneck more precisely than had ever been possible before. The cause of the bottleneck was multiple overlapping catastrophes. The Black Death massacres of 1348 and 1349, of which the Erfurt pogrom was one instance, wiped out large portions of the Ashkenazi population across Central Europe. Subsequent expulsions, forced relocations, and periods of confined ghetto living further reduced population numbers and limited genetic exchange with outside groups. The surviving communities were small, isolated, and highly endogamous, marrying primarily within the community for religious and social reasons. The same bottleneck that created unusual genetic uniformity also concentrated certain genetic variants at unexpectedly high frequencies. This is the origin of several genetic conditions that appear at elevated rates in the Ashkenazi population, including Tay-Sachs disease, Gaucher disease, and BRCA gene variants associated with elevated breast and ovarian cancer risk. These are not features of the original Middle Eastern or European ancestry. They are the statistical consequence of extreme founder effects. Random genetic variants that happen to be present in the small founding group and became common simply because everyone alive today descends from that group. To understand the genetic history of the Ashkenazi people, you also have to understand the social world that shaped it. Because, for most of the medieval and early modern period, that social world was defined by a single institution, the shtetl. The word shtetl is Yiddish for a small town. From roughly the 15th century onward, as Jewish communities were expelled from Western European cities, England in 1290, France repeatedly through the 14th century, Spain and Portugal in 1492. The center of Ashkenazi Jewish life shifted eastward into Poland, Lithuania, and the Russian Empire, where Jewish settlement was tolerated, if rarely welcomed. The shtetls were small, self-contained communities. They had their own religious courts, their own schools, their own charitable organizations, their own cultural life. They operated within the surrounding Gentile society economically. Jews served as merchants, innkeepers, craftspeople, and moneylenders, but remained socially and biologically separate from it. Marriage between Jews and non-Jews was prohibited by Jewish religious law and frequently by civil law as well. This social structure had direct genetic consequences. A community that marries internally, generation after generation, across hundreds of years, is a community whose genetic pool becomes progressively more homogeneous. Every child born in a shtetl shared more ancestors with every other child in that shtetl than would be true in a more open population. The bottleneck that had already compressed the founding population was sustained and reinforced by the social architecture of shtetl life. The shtetl also gave the Ashkenazi population something that the genetic record can partially recover: a distinctive dietary and health profile. Isotope analysis of bone chemistry from medieval Jewish burials, including at Erfurt, can identify broad patterns in diet from the chemical signatures left in bone by long-term food consumption. The medieval Ashkenazi diet appears to have included significant quantities of fish, particularly freshwater fish from the rivers and lakes of Central and Eastern Europe, along with grains, legumes, and dairy products consistent with kosher dietary restrictions that prohibited the mixing of meat and milk and placed specific restrictions on permissible animal species. The Yiddish language, the vernacular of the shtetl world, is itself a genetic metaphor. It is a fusion language, combining a German grammatical and phonological base with substantial Hebrew and Aramaic religious vocabulary, and significant Slavic borrowings acquired as the community moved eastward. It is neither German, nor Hebrew, nor Slavic, but a distinct synthesis created by a community that lived between worlds, drew from all of them, and belonged fully to none. The largest migration in Ashkenazi history was not the ancient diaspora from the Middle East. It was a movement that happened within Europe itself, between roughly the 14th and 17th centuries, as Jewish communities expelled from Western and Central European cities relocated eastward into the Polish-Lithuanian Commonwealth and later into the expanding Russian Empire. This eastward migration was, in historical terms, one of the most consequential voluntary population movements in medieval and early modern Europe. The Polish-Lithuanian Commonwealth, which at its height covered an enormous territory across modern Poland, Lithuania, Latvia, Belarus, and Ukraine, maintained a policy of relative tolerance toward Jewish settlement that was unusual in the European context. Jewish communities were permitted to own property, practice their trades, and maintain their religious institutions with a degree of autonomy that was not available to them further west. The result was a dramatic demographic expansion. The Ashkenazi population, which had been compressed by the catastrophes of the 14th century, began growing again rapidly in the more hospitable environment of Eastern Europe. By the 17th century, Poland had become home to the largest concentration of Jews anywhere in the world. Ancient DNA from burial sites along major river trade routes in Central Europe has identified individuals with Ashkenazi genetic profiles in commercial contexts. People buried alongside the trade goods of their profession, in locations that place them on the major commercial arteries connecting East and West. These individuals document the economic integration of Ashkenazi merchants into the broader European trading system, even as they maintained their social and genetic separation from the surrounding population. The genetic data from this period also shows the Slavic admixture that was accumulated during the centuries of Eastern European settlement. While the Ashkenazi community maintains strong endogamy as a cultural practice, no human community is perfectly closed. The Erfurt study and subsequent analyses of medieval and early modern Ashkenazi DNA show a small but detectable increase in Slavic genetic ancestry in Eastern European Jewish populations, compared to their Central European predecessors. The biological trace of generations of proximity and occasional admixture with Polish, Ukrainian, Lithuanian, and Russian neighbors. The community that arrived in Eastern Europe carried a genetic profile shaped by Middle Eastern origins, Italian maternal founding, and Central European centuries. The community that emerged from Eastern Europe centuries later carried all of that, plus a light signature of the Slavic world that had surrounded it. No aspect of Ashkenazi genetics has generated more controversy or more careful scientific qualification than the question of whether the population's documented high performance in fields requiring abstract reasoning reflects anything in the biology of the genome. The observation itself is statistical and factual. Ashkenazi Jews have won a disproportionate number of Nobel Prizes relative to their share of the global population. They are overrepresented in academic faculties, in the professions, in the arts, and in domains from chess grandmasters to Silicon Valley founders. The disproportion is large enough that it demands some kind of explanation. Two broad categories of explanation exist, and most serious researchers today believe the full answer involves both. The cultural explanation emphasizes that Jewish tradition has placed extraordinary value on literacy and learning for over 2,000 years. The obligation to study religious texts, to be able to read, interpret, and debate the Torah and Talmud, meant that Jewish communities maintained near universal male literacy at a time when literacy rates in the surrounding Gentile population were typically in the single digits. A community that prizes learning, that structures its social hierarchies in part around intellectual achievement, and that marries its daughters to scholars rather than warriors, creates strong cultural incentives for intellectual development across every generation. The genetic explanation, developed most systematically by Gregory Cochrane, Jason Hardy, and Henry Harpending in a 2005 paper, proposes that the social restrictions of medieval European life, which confined most Jews to occupations requiring literacy, numeracy, and abstract reasoning skills, created selection pressure over many generations that favored genetic variants associated with enhanced cognitive performance. On this account, the bottleneck that created genetic uniformity also concentrated variants that had been selected for by centuries of occupational restriction. This hypothesis is scientifically controversial and remains unproven. The genetic variants that would need to be identified and validated have not been conclusively linked to cognitive performance in ways that would confirm the hypothesis. What is not controversial is the cultural dimension. A community that has valued scholarship for 2,000 years has built extraordinary educational and intellectual institutions, and those institutions produce outcomes that are not surprising given their quality and intensity. The honest scientific position is that both factors probably contribute, that the relative weight of each is unknown, and that any claim of certainty in either direction goes beyond what the evidence currently supports. The 20th century imposed a catastrophe on the Ashkenazi genetic record that no previous historical event had approached in scale. Between 1941 and 1945, approximately 6 million Jewish people were systematically killed across German-occupied Europe. The majority of those killed were Ashkenazi Jews from Poland, Ukraine, the Baltic states, and Germany itself. The pre-war Ashkenazi population of Europe had been approximately 9 million people. By 1945, it had been reduced to approximately 3 million, and many of those survivors quickly left Europe for Israel, the United States, or other destinations. The genetic consequences of this destruction are measurable. Every person who was killed was a carrier of genetic variants that died with them. Entire family lineages, entire branches of the Ashkenazi genetic tree were cut off and permanently lost. The diversity that had already been compressed by the medieval bottleneck was compressed further still by the violence of the 20th century. In the decades following the war, the practical applications of DNA technology have created something that no previous generation could have imagined: the ability to find family connections across the ruptures of history. Ashkenazi families that were separated during the war, parents from children, siblings from siblings, cousins from cousins, have in some cases been reunited across generations through commercial DNA testing, when descendants on different continents discovered unexpectedly high genetic matches in testing databases. The story of one such reconnection, documented by researchers working with survivor communities, involves a family separated in 1942 in Warsaw. The grandmother survived and reached the United States. Her grandchildren, 3 generations later, took a commercial DNA test for curiosity. The test returned an unexpected high-confidence match to a family in Israel. Research confirmed that the Israeli family were the descendants of the grandmother's brother, who had escaped to Palestine before the war. 2 branches of a family, separated for 80 years, reconnected by the match of 4 letters in a strand of ancient molecular code. The Erfurt study, and the broader scientific program it represents, is not only about the medieval past. It is about understanding a genetic community that has been repeatedly compressed, displaced, and partially destroyed, and that has, repeatedly, survived. Let's bring the entire story together, because the Ashkenazi DNA investigation is not just the story of one community. It is a case study in what DNA science can tell us about the universal human experience of movement, survival, identity, and change. The genetic record of the Ashkenazi Jews documents one of the most complete and traceable population journeys in the ancient DNA archive. It begins in the Levant, in the hills and valleys of modern Israel and Palestine, where the ancient Israelite population lived, developed a distinctive religious culture, and over centuries came under the successive control of Assyria, Babylon, Persia, Greece, and finally Rome. The Roman destruction of the Second Temple in 70 AD and the subsequent suppression of the Bar Kokhba revolt in 135 AD triggered the major westward diaspora. Jewish men moved into the cities of the Roman Empire, Rome itself, the cities of North Africa, the ports of Spain and southern France. In Rome and in Italy, some of those men formed families with local women who converted to Judaism. The mitochondrial DNA of those Italian women entered the community and has been carried forward to the present day, confirmed by the Erfurt ancient DNA analysis. The community that formed in medieval Central Europe, the community whose cemetery was sealed in Erfurt in 1349, was already a synthesis. Middle Eastern in paternal ancestry, Southern European in maternal ancestry, unified by a religious and cultural framework of extraordinary coherence and durability. The Erfurt DNA confirmed that synthesis directly from the bones of real people who had lived, worked, prayed, and died in a medieval German city before a night of violence sealed their remains underground for 7 centuries. The bottleneck of the 14th century and the social architecture of the shtetl communities that followed it created the unusually uniform genetic profile that modern Ashkenazi individuals share. A profile so distinctive that genetic testing companies can identify Ashkenazi ancestry with high confidence from a saliva sample. That uniformity was not a design. It was the biological consequence of persecution, isolation, and the random survival of a small group of people whose descendants multiplied into millions. The 20th century catastrophe imposed a further compression on a genome that had already been compressed. And yet, the community survived, dispersed now across Israel, the United States, Europe, and the rest of the world, carrying a genetic signature that is among the most studied and most precisely characterized in the entire field of population genetics. What does the Ashkenazi DNA story teach us that goes beyond the specific community? It teaches us that identity is never simple. The Ashkenazi genome is Middle Eastern and European simultaneously, neither fully one nor fully the other, but a specific synthesis created by a specific history. There is no pure origin. There is no bloodline that arrived from a single point and stayed unchanged. Every human population that has ever been studied shows the same pattern of mixture, movement, and layering. It teaches us that culture is a genetic force. The social structures that maintained Ashkenazi endogamy, religious law, community institutions, the physical architecture of the ghetto and the shtetl, shaped the genome as surely as any migration or conquest. What people choose to do across enough generations leaves a biological mark. And it teaches us that the genome is a historical record more durable than any archive, more honest than any chronicle, and more intimate than any biography. The people buried in Erfurt in 1349 left no names that survived. Their individual stories are gone, but their DNA remained, sealed in bone, waiting for a technology that did not yet exist to read what they could not say. History is written in the genome, and the genome does not lie.